Hello, viewers. Thank you once again for joining Alpha One Canada's 2015 Virtual Education Summit. My name is Duncan Lamb, and I'm your host. We're very happy to have our speaker here today, Dr. Stephen Roblin from Grifols Canada. Dr. Roblin will be speaking to us about plasma safety and the manufacturing of alpha-1 antitrypsin augmentation therapy. Dr. Roblin, I'll let you say a few words to our viewers before we begin. Do you mind sharing with us your experience and areas of expertise?
Sure, Duncan. I guess before I do that, maybe I'll just thank you guys for giving me the opportunity to participate in this, what I know is going to be a very fantastic initiative. I guess present company accepted, you have lined up some very, very good speakers. And, just personally, I'm really looking forward to hearing, what they had to say around Alpha-1. My experience and expertise, I really work in rare diseases and diseases that are treatable with plasma-derived therapies. That's really going to be the focus of the talk. I do a lot of collaboration with scientists and researchers around trying to generate and develop new products to treat these diseases, making our current products more safe and effective. And I also do a lot of education, so I'm excited to participate. I hope that the talk is interesting to questions when we do the Q&A in a week or so, I guess.
Thank you very much, Dr. Roblin. Well, without further ado, let's proceed to today's featured presentation.
All right, so as was mentioned in my introduction from Duncan, my name is Dr. Stephen Roblin. I work in the Medical Affairs Department for a pharmaceutical company called Grifols. Part of my role is in education related to the diseases and therapies where Grifols has a specific focus. So since other presenters in this great virtual summit are going to be discussing Alpha-1 as a disease, as well as different strategies for living and coping with the disease, for managing the disease, including therapy options like augmentation therapy, I'm actually going to take a little bit of a different and unique approach to a topic related to Alpha-1 that's actually a really important topic related to the therapy. And that's something called plasma safety, which many of you may not be that familiar with. So before we start into the presentation, I wanted to give you a little bit of background into Grifols as a company. And interestingly enough, Grifols just celebrated its 75th anniversary and has really been working in the healthcare arena since around the 1940s at the start of the Second World War. Grifols is actually a very unique type of pharmaceutical company. It's a company that's primarily devoted to what we call plasma-derived therapies. And these are generally therapies that are used to treat very rare, chronic, and life-threatening conditions, such as alpha-1 antitrypsin deficiency. But there are other therapies which we're not really going to focus on today to treat hematological conditions, such as hemophilia or bleeding disorders, immunological conditions, such as antibody or immune deficiencies, or autoimmune diseases, things like infectious diseases, as well as patients who become burned or have had traumatic injuries. So this is the presentation overview. We're going to start with what we call plasma donation, and this is really the donation of the raw material, which is plasma, that we use to manufacture all of our biological therapies, including alpha-1 antitrypsin augmentation therapy. We're then going to move on to what we call pathogen safety or plasma safety. This is really related to making sure that the road from plasma donation all the way to that medicine getting into the patient is safe. And then finally, we'll talk a little bit about manufacturing, since that's a very important aspect of plasma safety and purification of these medicines from plasma all the way to a concentrated medicine that's used to treat a very specific disease. I'm going to put this slide up really to give a bird's eye view of this entire process, because I know this is something that many of you are going to be completely unfamiliar with. So what we'll do is go stepwise through each of the main areas we're going to talk about, and then within the rest of the presentation, I'll focus on each of these areas a little bit more specifically to give a little bit more detail so people can get a better view of what we're talking about. So the process really starts with donor qualification and selection, and then collection of their plasma through a process called plasmapheresis. We then move on to testing that plasma primarily for a battery of pathogens that we certainly wouldn't want to have moving into the manufacturing process. Then we have the manufacturing process itself, which is we basically separate into something we call fractionation and purification. But what that really means is taking all of the different proteins in the plasma and separating them out and purifying them out into very specific molecules or groups of molecules or proteins or medicines that can be used to treat patients with these rare diseases. And what's really interesting about this is the time it takes from a donor walking into a plasma donation center all the way through to a protein that they have donated becoming a specific medical product takes up to a year. So as I mentioned, we'll start out with this aspect of what we call plasma donation. We'll talk about Grifols' plasma donation philosophy, and then we're really going to focus on two things. One is this idea of donor screening and testing to make sure that the plasma that we get from these donors is as safe as it possibly can be, but at the same time, the individuals who are donating plasma, we want to make sure that their health and well-being is maintained, because certainly we want to have that experience be positive and to have them continue to come and donate the raw materials for these life-saving medicines. Grifols has a very simple and straightforward philosophy related to plasma, and that's a commitment to donor health as well as the safety of the plasma that these donors have generously provided to make sure it's as safe as possible when it is administered to a patient who needs it for a disease. So I've been using this term plasma. So what is plasma? Well, plasma is really, if you want to break it down, it's the liquid portion of your blood that contains all of the proteins, all of the enzymes, all of the antibodies that make the majority of the biological systems in your body function correctly. There are other parts of the blood, like the red blood cells, which is actually the part of the blood that makes the blood a red color. That part of the blood is responsible for transporting oxygen and carbon dioxide within the body. But the plasma is really the part that we are definitely focused on because, as I mentioned, that's where the proteins are. And the proteins are what can be extracted and purified and used in medicine to help treat disease. Now that we've established that the plasma is where the proteins are, and specifically where the alpha-1 antitrypsin protein is that we would like to purify into a medicine, we have to collect the plasma. And that collection is done through a process called plasmapheresis. And although this is relatively complicated from a technical perspective, from a conceptual perspective, it's actually quite simple. I'm sure that everyone is aware of what a blood donation is. So this is very similar to a blood donation in that a donor donates blood, but instead of that blood going into a bag like you would have for a whole blood donation, it goes into a machine. And within that machine, the blood is separated into its different components, one of them being the plasma, which is collected. But what's very interesting is the red cells can be given back to the donor. And that's important because it allows donations to happen more frequently than they would if just whole blood was collected from the donor. Because red cells regenerate much more slowly than the proteins within the plasma. So where is this plasma actually collected? Well, it's actually collected from plasma collection centers. So these are centers that Grifols owns and operates in the US, and there's more than 150 dedicated plasma collection centers. And what's amazing is that across all of these centers, we collect about 25,000 plasma donations every single day. As I'm sure you can appreciate after seeing that map, over 150 plasma centers, 25,000 plasma donations every day. This kind of operation needs very, very tight oversight and is required to meet very strict safety and quality regulations and guidances from regulators, government regulators across the world, not just the FDA, which is the agency that's responsible for the US, which is where the sites are, but also from Health Canada, because the plasma that's collected from these facilities is ultimately going to be used in medicines that are used by Canadian patients. And this kind of global quality and safety framework is extremely important to ensuring that the plasma we collect is as safe as it possibly can be. I'm going to walk you stepwise through the plasma donation process. From the moment a donor walks through the door of a plasma donation center, all the way through to the completion of their donation, there are a variety of safeguards that are put in place not only to ensure the health of the donor, but also to the safety of the product, which I mentioned previously. We'll talk about what the experience is for a brand new donor, how that donor registered, and then effectively how we measure the risk of that donor from the perspective of, do we want to accept their plasma for donation? Do we want to qualify that donor? And that's done through medical assessments. It's done through risk evaluation questionnaires. And then once that patient is actually qualified, what is their experience in terms of giving that plasma donation? So let's start off with the new donor. Grifols relies on source plasma, which again is plasma from plasmapheresis, from repeat donors for continued monitoring of donor health and plasma safety. So prior to the use of their plasma for the production of these medical therapies, plasma donors must undergo, number one, a complete medical examination, number two completely separate health history screens, and number three, 2 sets of infectious disease screening tests. Plasma donors who meet these requirements are known as qualified donors, resulting in regular committed plasma donors. The process starts with donor registration. Steps in the donor registration process are designed to help ensure the donor is a part of the local community and that is actually safe and suitable for him or her to make a donation. This process involves identification of their ID, validation that they have a suitable and permanent address, and very importantly, cross-checks with what we call the National Database of Deferred Donors. And this is an industry-wide database where different organizations or companies that collect plasma share information on their donor lists, which allow them to understand if a patient has been disqualified or deferred from one plasma donation center, that they don't walk across the street or go to another area to donate plasma at a different center, and that there's that coordination. Now that a donor has been registered, the next step is the medical assessment. And this is a very comprehensive assessment that includes physical exam, measurement of vital signs, measurement of weight, various measurements that predict general health, as well as a donor education program that gives them information on not only the importance of their health as a donor, but also the uses of the plasma that they're generously donating to medicines related to various diseases, including alpha-1 antitrypsin deficiency. In addition to the comprehensive physical and medical testing assessment that I talked about on the last slide, plasma donors must complete a personal health history questionnaire. And the purpose of this is to give another aspect to the risk assessment testing that we're doing at Grifols to understand if a donor is suitable to donate plasma. And this questionnaire really involves asking them questions around their travel history, medical history, social history, any symptoms of illnesses that they may have had in the past or currently have, and their medications. And again, the idea here is to generate a risk assessment profile from that patient to decide whether or not it's appropriate and suitable for them to move forward in the process in terms of donor qualification and actually donating plasma. Assuming that the donor has met the qualifications from the medical and physical exam perspective, as well as from the risk assessment questionnaire, they can now move into the process related to plasma donation itself. So the entire process takes anywhere between 60 and 90 minutes. And as they're giving their donation, the donor is very carefully monitored by very well-trained and qualified staff at the donation centers. What's interesting is that a donor actually has to donate at least 2 times before their plasma can be used to produce therapies. And I'll talk a little bit about that later. A donation can be made no more than 2 times in a 7-day period. And this is primarily related to maintaining the health of the donor and as well as the quality of the plasma, because it does take a certain amount of time for all of the proteins to be regenerated within the body. And because these donors may be donating quite frequently and donating certainly their time, they are moderately compensated for their time and commitment to the donation process. Every donation is held for a minimum of 60 days before it's actually used in the manufacturing process to start purifying it into these individual medicines. The purpose of this is to allow us to address what we call the window period. And it's kind of a complex concept, but really what it means is that period between, and everyone can kind of think of this, I think, when we're thinking about when we get sick with a cold, right? Is that you might start feeling a cold, but you don't really know you have one. And there's that period where you're probably contagious, but you don't really know you have the cold. And then 3 or 4 days later, you start feeling sick and you know that you have that cold. And probably everyone else around you knows as well. This is very similar to what we're looking at from this window period in more serious infectious diseases, where what we don't want to have happen is for someone to come in at the very early stages of having an infectious disease, not knowing they have it, not having any symptoms, and making it through the qualification process and actually donating plasma, and then that plasma going into manufacturing. So by having this 60 days in between, and particularly from having this requirement for a person to donate plasma twice, separated by the 60 days, if they're a first-time donor, is we minimize this window period, and it's much more likely that we're going to be able to detect a pathogenic disease because we've had this additional 60 days of time to allow it to develop and to allow it to be able to be detected from our various testing methods, which I'll talk about. On that note, we'll move on to pathogen safety. We'll talk a little bit about the testing labs that we have at Grifols. We'll talk about how we test, and we'll talk about the various inactivation steps that we actually have in our manufacturing processes. Before I get into more details relating to pathogen safety testing and how we eliminate pathogens and viruses within our manufacturing process, it's really important to take a step back and reiterate that every single step in this process matters. Our mission every day is to ensure the safety of Grifols products from existing and potential pathogens. With this mission, we are completely open and transparent about the real risks associated with all plasma-derived products. And the reality is that Grifols products are made from human plasma. Because of this, no matter how many processes and procedures and regulations we follow to make sure our products are as safe as we can possibly make them, there's always a, although very small, potential risk to transmit infectious agents, such as viruses and theoretically prion disease, also known as Creutzfeldt-Jakob disease. We can't completely eliminate this risk. There's also certainly a possibility that unknown infectious agents may be present in plasma-derived products. But what I want to present to you in the following slides is everything that we're doing to try to make this risk as low as technologically possible, and to also talk about, or at least from a conceptual perspective, talk about how we are continually trying to improve our processes with new technologies and new ways of doing things to make that risk even lower. So now that we have the plasma donation, we've talked about that whole donation process. The next step is actually testing that plasma. And that's what we call donation testing. So each plasma donation undergoes rigorous pathogen detection and testing. And again, I mentioned we have 150 donor centers, 25,000 samples or plasma units that we need to test every day. And with a minimum of 10 tests per donation, if you do the math, this adds up to a very staggering number, which is 52 million tests for pathogen safety that are done every year. And this is done at 2 different laboratories, one in San Marcos and one in Austin, Texas. So as I mentioned, plasma donations undergo a series of tests. This state-of-the-art testing helps ensure donor health and safety of plasma donations for the use of their manufacturing medicines. And this includes testing for the following pathogens and donor health markers. So every single plasma donation, every single bottle of plasma is tested for HIV, hepatitis A, B, and C, as well as parvovirus B19. We also have periodic testing related to liver function, serum protein electrophoresis, blood cell antibodies, as well as syphilis. I want to give you a very brief description of these tests that we're using to detect the presence of pathogens within the plasma donations. And we use effectively two different technologies. We use something called nucleic acid amplification technology, or NAT testing, and we use something called serology testing. And really they're designed to detect two different things. NAT testing is really designed to detect the DNA of the virus itself. So this is really focused on, is there any virus in the plasma? Very important test, very sensitive test. It's a newer, more recent technological advancement in testing. But we also have something called serology testing. And serology testing is a little bit different. It actually detects proteins on the virus, or it detects the body's response to a virus. So if someone's had a previous exposure to a virus, then they will have developed antibodies towards that virus, and we can actually detect those antibodies. So it gives you a little bit of a different way of getting to the same goal, which is detecting, is there any virus in the plasma, or has the person that donated that plasma been exposed to that virus in the past? And this allows us to, again, potentially, if we get a positive test on one of these, go back and pull that plasma unit, which hasn't gone into the manufacturing yet because it's been waiting on that 60-day hold that I talked about, and make sure that unit is pulled and doesn't go into manufacturing and destroy that unit. What would also happen is if that donor was positive for HIV or one of these serious viral illnesses, that they would be contacted, and they'll also be put on that national deferral donor database that I talked about previously to effectively dequalify them or disqualify them from donating plasma in the future. So we've done all of this work to screen out pathogens from making their way into the manufacturing process. We've done that through the donor screening, which I talked about, the medical examinations, the risk examinations, the individual testing of the plasma units, the testing of the manufacturing pools. So we've done as much as we can to try to make sure that virus didn't get anywhere near the manufacturing process. But as you can imagine, we do not test for every single possible virus that we know about. And certainly it's not possible to test for viruses that we don't know about, what we would call emerging pathogens. So because of that, the actual manufacturing process itself has been designed to have multiple steps to either inactivate or remove all different types of viruses. And this is a very, very complex procedure, and to be perfectly honest, I could probably give another hour of this presentation just devoted to these manufacturing steps and how they eliminate different types of viruses of different sizes, of different shapes, different types. But I think the main thing is just to understand that there are these multiple steps, that the amount of virus that is required for these steps to eliminate is very heavily regulated by the same health authorities that I talked about before, and that we are constantly, as I mentioned, looking at ways to add new steps in if we need to further increase what we would call the margin of safety of the product. To follow on with this concept of trying to maintain knowledge about new pathogens that we may have to be concerned about related to our pathogen safety program, Grifols maintains a very, very close working relationship and has defined and established lines of communication with leading health authorities around the world. And these include people like, or agencies like the Centers for Disease Control, European Medical Agency, the FDA, Health Canada, and others. And the purpose of this is to have effectively ongoing vigilance around the emerging and development of new pathogens and giving ourselves an opportunity to react as quickly as possible if we need to. For the last few slides in this presentation, I'm going to overview the manufacturing process in general, but more specifically related to the manufacturing and purification of alpha-1 antitrypsin. So I'm going to give you a little overview of manufacturing facilities that Grifols operates. And you're probably wondering, why are manufacturing facilities important? Well, they're important because, as you can, I'm sure, imagine based on the slides I've gone over so far, the manufacturing of these products is very, very complicated, given we have to collect plasma, we have to test the plasma, we have to manufacture the plasma. And what we want to make sure is that we have the flexibility and the capacity to ensure supply and manufacturing of these medicines for patients. And that's really essential to maintaining access to these products. And that's why it is important to have multiple manufacturing facilities around the world. So if we go way back to the beginning of this presentation, I mentioned these two different processes that are involved in separating out the proteins in the plasma all the way down into their individual proteins. And these are two processes, and we've separated them and named them different things. We call the first part fractionation, and we call the second part purification. But really, they're pretty similar in that they're really just these separation techniques. And I'm not going to go through the complexities here or go through these diagrams, other than just to sort of, from a conceptual perspective, tell you that what we're doing is we're taking all of these plasma donations and we're pooling them into these very large pools of plasma, thousands and thousands of liters, and we start to separate those proteins out from one another into groups. And these groups are called fractions, and that's why it's called plasma fractionation. And what we do is we further purify them down into the individual proteins. So we have what we call factor products. And these are proteins that are important in the coagulation of blood. So people that have bleeding disorders, that they're missing these factors. Many people will have heard the term hemophilia. These are the people that are missing these particular proteins that are in plasma, and we're purifying them out to give to those people to help them with their bleeding disorder. We also have a group of proteins called immune globulins. Immune globulins are proteins that are basically antibodies. So this is a whole bunch of antibodies from all of these different donors, and they're used to treat people that have a deficiency in antibodies or immune deficiencies, but they're also used to treat people that have an overactivity in their immune system called an autoimmune disease. And for a variety of reasons, these purified antibodies seem to help those patients with those autoimmune type diseases. We also have, and I'm sure what most of the people listening to this video are interested in, alpha-1 protease inhibitor or alpha-1 antitrypsin. And then we also have other proteins called antithrombin, which again is involved in coagulation, bleeding, and then albumin, which is used in a variety of different areas in medicines. But one of the areas is in things like burns, sepsis, and trauma. I'll very briefly go over some of these specific manufacturing details for alpha-1 antitrypsin augmentation therapy, also known as alpha-1 protease inhibitor. I don't think it makes a lot of sense to go over all of these steps in detail and talk about the biochemistry and the science behind each one of these processes. But it is, I think, a little bit interesting to see this manufacturing process in a diagram and as an overview, and I'll just highlight a couple things that I think are maybe interesting, and then what we'll do is go through in the next couple slides some actual pictures of the manufacturing processes related to these different steps. And what I want to highlight, and I think again, since this is a plasma safety talk, I've highlighted the areas where we have either a specific virus or prion disease reduction step in the process, and whether we have a virus reduction step in the process. And I think this just shows you from that big list of different processes and procedures we have to eliminate viruses and pathogens from our manufacturing process, this is the specific part of the process related to alpha-1, where in addition to helping purify and make the product really specific for treating alpha-1, also is involved in these viral inactivation and reduction steps. So these are things like what we call PEG precipitation, depth filtration, solvent detergent incubation, and then nanofiltration. And again, without getting into the details, I think it's just important to think about some of these things and at least have an idea of what this means. Here are some interesting pictures of different parts of the manufacturing process. And these are really specific pictures. These manufacturing plants are absolutely enormous. But we've taken some pictures and put them up here just to give you an idea of what this looks like in real life. And again, without going into the names of these things and what they mean, I'll just sort of flip through some of these things and talk a little bit about them. So we've got earlier in the process, we have this paste suspension part, which is part of the fractionation process. And then we have this part called solvent detergent treatment, which again is a specific process that's related to viral inactivation, but also is involved a little bit in some of the purification of the process. We have sort of two different things happening that are important in the manufacturing. Moving on to this next slide of pictures, we have anion and cation chromatography. And chromatography is just a fancy scientific word for separating something from something else. So really what we're talking about here is using the charge of these different proteins, whether it's a positive charge or whether it's a negative charge, using that property of those proteins to separate them out from one another, which is a pretty neat way of doing things. And then we also have something called ultrafiltration and difiltration, which is a way of further purifying and concentrating the product down, which is valuable in making sure the more concentrated the product is and the less impurities it has, the easier it is to administer, the faster it is to administer, the lower volume it can be reconstituted in, and that's really beneficial from, I think, a patient perspective. This is the final slide in the presentation. I really wanted to come full circle, and we started out this presentation talking about what plasma is, what proteins are in plasma, how the donation process happens for plasma, the screening, the medical examinations, the donation testing, the manufacturing, and how we get from that donation all the way down into these individual proteins that are really required to treat patients with all sorts of different diseases. And I think the best way to end this is to come all the way back to say it is really amazing how much plasma is actually needed to treat a patient, the average patient, with one of these conditions listed here just for 1 year. And remember, many of these conditions are chronic and may require treatment for life. So for a patient with an immune deficiency, which we had talked about before, it actually takes 130 individual plasma donations to treat that patient for a year. A patient with alpha-1, 900 donations. And a patient with hemophilia, 1,200 donations. So we really have to, I think, all thank the plasma donors for providing this very generous resource and in giving and donating this plasma to allow the treatment of patients with these this diseases. So this wraps up my presentation. I'm glad to have been able to go through this interesting aspect of these plasma-derived therapies with you. I hope that everyone's at least learned something. And if anyone has any additional questions, I hope that I'll have an opportunity to answer them in the question and answer session that's being planned within a few weeks of this recording. And if not, then I'm certainly always able to be reached at Grifols, and I think anyone that needs to reach me can probably get my information through the Alpha One Canada Foundation. Thanks very much.
This concludes today's featured presentation. On behalf of Alpha One Canada, I would like to thank Dr. Stephen Roblin for taking the time to prepare and speak to our viewers. I think it's really reassuring for patients to know more about the treatment that are available to them. Please don't forget to check out the rest of our 2015 Virtual Education Summit featured videos on our website, www.alpha1canada.ca. Thank you very much and have a wonderful day.